Yes. Dear students, welcome to the another live class of Edwins. Please do hurry and join the live class so that we can start our live session. Please do hurry and join the live class so that we can start the live session. And before we dive into today's session, make sure to subscribe our YouTube channel, Edwins, if you have not already yet, so that you can never miss the live classes and new educational videos. Also, an important announcement for the board students of West Bengal. You may visit our website and purchase the mock test as well as MCQ test series to betterment of your exam preparation. So please do visit our website edwins.in and do prepare yourself through the mock test and MCQ test. Please do hurry and join the live class so that we can start the live session. A very good evening to all of you and welcome to the another live class of Edwins. Hope you spent a wonderful and enjoyable puja vacation, and now it's time for study because our text exam is coming soon. So for your preparation, our Edwins has recently launched the free mock test, free MCQ test series. Yes, it's free MCQ test series and paid mock test series. So please do visit our website edwins.in and get prepare yourself for the Madhyamik exam. So let's get into the topic of today's discussion. Today we will discuss about the heredity and some common genetic diseases. From this chapter we will discuss about the heredity and Mendel's experiment. So now first we have to know that what is heredity. What is heredity? We know the heredity is the passing of characters from one generation to another generation. What is this? This is if this is a parent, like your father or your mother, that is the eye color, a character. Eye color is blue or brown. It will pass to you and offspring, to the offspring, that is you, and the eye color is blue or brown. Okay, so this is called the heredity, and another term is inheritance. What is inheritance? Inheritance is the phenomenon of passing those characters. Okay, so this is the inheritance and heredity. But one question, that is, if my father or mother contain blue or brown eyes, but that is my eye color or my brother's eye color is not same. It is happening. So why this is happening? This is happens due to the variation. Now what is variation? Variation is the number 2, variation. What is variation? Variation is the changing of characters from the parents, or this is can be different from the parents or from the siblings. Like this is you, this is your brother or sister, anything. So your eye color is blue, but your brother eye color is brown. This is called the variation. That is eye color blue, but your brother or sister eye color is brown. This is called the variation. Variation is the characteristics, one important characteristics in any population, and this is specially occurs in the human. This is very much important. That's why we are not look like as similar as to our parents or our siblings. So this is occurs due to the variation. Then what is the cause of this variation? Of course, the question is the cause of the variation. Why the variation is happening? That is cause of the variations. Number 1, of course, in cell division we have already learned, that is in sexual reproduction the male and female gamete, the haploid gametes contain half number of chromosomes. During cell division, these chromosomes form the chiasma, build the chiasma and exchange the genetic materials. Exchange the genetic materials. Okay, so by exchanging these genetic materials, this is occurs in meiosis. Okay, exchanging the genetic materials occurs the variations in human or others organisms. Okay, first cause. Second one, that is first is the meiosis division. Second one is the mutation. Sorry. Mutation. What is mutation? Mutation is the changing of genetic sequences What is genetic sequence? New term, of course. Genetic sequence is nothing but changing in DNA. We know the basic unit of genetics or gene is the DNA. There, a specific genetic sequence or DNA sequence followed in every organism. But when any external or internal causes, like the external cause, that is radiation, due to radiation, this part is mutated. Okay, of course, this part too, this part of the DNA is get mutated. Okay, so this is happens and causing the variation in organisms. Always this mutation is not negative or positive. It can be negative, it can be negative, it can be positive. How? Example, negative, of course, we all know, that is cancer. In case of positive, that is in any changing environment, in any changing environment, the organism get mutated, and which is beneficial for its survival. Like in suppose a cold or a desert animal migrate to a older region. Okay, then it will get mutated itself, and it get mutated itself for survival in changing environment. Okay, so this is the variation. Okay, one of our students asked that the difference between mutation and variation. Of course, the mutation that is mutation and variation. What is mutation? Mutation is the result of, mutation is the cause of the variation. How? A gene sequence or a DNA sequence is get mutated. Okay, this part get mutated. For this mutation, the variation is occurs. How? An example, that is like this is a gene for the skin color. Okay, this is for bright skin color. Okay, and the animal is or the human living in the polar region. Okay, now he or she will migrate to the equator region. He or she will migrate to the equator region. Then the gene of this bright skin tone is get mutated, and it will get for the dark skin tone, which will help to absorb sunlight, because in equator region, of course, the sunlight is very high, which will help to absorb the sunlight. So this is the difference between mutation and variation. Mutation is the cause and variation is the result. Now, before we going to the Mendel's experiment, a few important terms we have to know. So what are they? First, the character and trait. This is very much important to understand those concepts. What is character? Character is the general form of identifying any species. Like example, character is the height. Okay, so the height will vary, that is tall or short. Okay, so this tall or short, this is denoted by traits. The traits will specify the character. Character is used in general, that is height or the eye color or the hair color, etc. But the trait will specify, that is the eye color is brown or black or blue. The hair color is dark, that is black or brown. These will specify by the traits. That is, character is used in general form, but the trait is used to specify the character. Okay. Now, the second is the allele and gene. Allele and gene are as like as the character and trait. How? Like this part of the DNA contain gene for the eye color, gene for eye color. Okay, like black eye color, or this can be made by this. The color is, this is also black and this is also black. Okay, but the allele will specify or denote the presence of the traits inside the gene. Like the capital B denoted for the black eye, but the small b denoted for the brown eye. Okay, the gene is here also, the gene is used for the generalized term, that is that denoted the character, but the allele will denoted the traits. Okay, so this is the difference between gene and allele. We always call that alleles are the alternative forms of gene. How? Because genes are used in general term, that is eye color or height or hair color, etc. But allele will specify these genes character, that is in form of the trait. Okay. Next, the term that is the locus. Locus. What is locus? Locus is derived from the word that is loci, which is mean place. So what is the locus? Locus is actually inside the DNA where the gene is located. Like this part of the DNA contain active genes. What are the genes? Of course, the functional part of the DNA. DNA contains some functional and non-functional parts. The functional parts of the DNA which can able to produce a specific protein. Okay, this part is known as gene. Okay, so this gene where inside the DNA that gene is located, this part of the DNA is called locus. Okay, locus is the place. This is the place where the gene is located. Okay, so this is the locus. Next, for the Mendel's experiment, a few terms that is very much important to learn. That is, number one is pure and hybrid. Pure and hybrid. What is pure and what is hybrid? Pure, that is contain same kind of allele. What is this? Like if for the black eye, this can be black and this can be black. Okay, but this, the number 1 and number 2, this capital B and capital V contain same kind of alleles. That's why this is called pure. But here in second, here contain a capital B and a small b, which is different. One is capital B, one is small b. So these are different. So this is called hybrid. Different forms of gene or different forms of allele present in a gene, this is called hybrid. Okay. So this is the pure and hybrid. So here we can observe that is capital B, small b, and capital B, and capital B, small b. From these, we previously discussed that capital B responsible for black and small b responsible for brown. But the brown character is not expressed when the capital B is present. From this, the next terms, that is dominant and recessive allele. What is this? The dominant allele is called those when it present in one. It can able to express its trait. When it is present in one, it can able to express its character. This is black. But recessive is the recessive allele is those where it can only express when it is present in pair. That is, it can only able to express when it is in pair. This is the brown. Okay. So this is the pure and hybrid, and it is how it is related to dominant and recessive. The next one is the phenotype and genotype. From here we can discuss. That is, next one is the phenotype and genotype. What is phenotype? Phenotype is the outer characters or observable characters. That is, here present capital B, here present capital B, small b. But in both cases, we can observe the black eye. This is called the phenotype. Then what is genotype? The types of alleles present inside it. When we analyze that types of genes or forms of genes, that is the traits present inside it, this is called genotype. The types of alleles present inside a gene, these are called genotype. That is, in case of this, there have two alleles, that is capital B, but here present two different types of alleles, that is capital B and small b. So this is the phenotype and genotype. Phenotype is the observable character and genotype is the alleles present inside a gene. Okay. Next one is the hybridization and the cross. First one is the monohybrid, the cross. That is, there are two types of cross. monohybrid and dihybrid. What are they? From the name, that is mono mean single, di mean double. That is, the cross is occurs by the single characters, only in single characters. Okay, like in Mendel's monohybrid cross, we will learn, that is, he took the height of the plant, This is the monohybrid cross. That is, in case of tall plant, that is capital T, capital T. In case of dwarf plant, this is small t, small t. Next, in case of dihybrid, here two different characters are taken. How? Here, this is the height of the plant. We can add, that is, color of the seed. Okay, so then it will be capital T for the height plant, and for the green seed is capital G, capital G. And in case of dwarf, small t, and in case of yellow seed, that is small g, small g. Here we can observe, this is the pure, this is also pure, and this is the dominant trait. Always the dominant traits are, or dominant alleles are written in capital, and the recessive one is the written as small. So this is the recessive trait. This is dominant. Okay, so this is the process of cross. Now the Mendel's experiment. What is this? Mendel was a monk of the church. He was very passionate about gardening. Okay, he was very passionate. So in his garden, he cultured the pea plant. Why pea plant? Because it is easy to access first, and it will develop, and the development and maturity of the pea plant is very fast. So he chose the pea plant. In monohybrid cross, he took the character, that is height of the plant. And he classified in two groups, that is the height or tall plant and the dwarf, that is tall and dwarf. In tall, it denoted capital T, and in case of dwarf, denoted as small t, small t. Then he start the crossing. So in crossing, that is capital T cross with the small t, small t. This is the parent generation, which is denoted as P1. Of course, from the parent generation, it will form the gametes. The gametes are capital T, small t, small t. So the possibilities of the offsprings is 1, 2, 3, and 4. So capital T, small t, capital T, small t, here also capital T, small t, and capital T, small t. So in first generation, sorry, the all offsprings he will get is tall, but they all are hybrid. This is called the heterozygotic condition or heterozygous. What is heterozygous? When two different types of allele present in a gene, this is called heterozygous. But in parent generation, he was took the homozygous. So this pure form is called homozygous and the hybrid form is called heterozygous. Okay, so this is the first generation, that is F1, filial generation 1. He will get the all tall hybrid plants. But in next generation, what he will do? He did the cross between the offsprings. Like how? In P2 generation, that is parent 2, this was the parent 1. P2 generation, he crossed between capital T small t and capital T small t. Okay, so the gametes capital T small t, here also capital T and small t. This combination of the cross was denoted by a board or box which is called the checkerboard or Punnett square. So what is this? In this, okay, so we will put the first one, that is capital T small t, and the second one, that is capital T small t. Here this is male, this is female. So how we will cross it? First, the combination capital T, here capital T, capital T. Next, that is capital T and small t, capital T, small t. In this line, here small t, here capital T, that is capital T, small t, and here small t and small t. So this is the ultimate result of the cross. Okay, and here he observed that in the second generation, that is in F2 generation, the 75% plants are tall, but 25% dwarf. From these, he put the phenotypic ratio. Phenotypic mean the observable character. So capital T, this is tall. Capital T, small t. Capital T is denoted for the tall trait, that is also tall. This is also tall, but this is short or dwarf. Okay. So the phenotypic ratio will be 1 to 3. 3:1. Okay. But the genotypic ratio is here, this is tall, that is 1, but these two are tall but not the homozygotic. These are the heterozygotic. So these two are 2, and the dwarf, the last one, is the 1. So there have two homozygotic, one is homozygous dominant, another one is homozygous recessive, and these two are the heterozygous. So the genotypic ratio is 1:2:1. So in monohybrid cross, that is the genotypic ratio is 1:2:1, and phenotypic ratio is 3:1. You have to remember this. What is the genotypic and what is the phenotypic ratio of monohybrid cross? So that's all for the today's live class. If you have any doubts, you may ask in our comment section. We will definitely discuss it in our next class. And if you like to get more such informative video and attend such live classes, you may subscribe our YouTube channel Edwins, and don't forget to prepare yourself for the Madhyamik exam. You can purchase our mock test series, and please do practice the free MCQ test in our website. You can visit our website edwins.in and enroll yourself. Thank you.